So I'm going to start off with my acknowledgements. So the majority of this work was funded through the MB Endeavour Beyond Myrtle Rust program, sort of shifting into the Naraki Takitaki project as well. But without earlier funding from the MB Catalyst and the MPI and the initial phases of the incursion, we wouldn't have been able to do the work that I'm presenting now. And there's again a bigger project team that underpins all of that work. And just to now place us back in New Zealand in Aotearoa. So you've heard already that a range of taonga species are affected, and work that we've done screening some of these species has shown that there's little to no resistance in Ramarama, Rohutu or Pahudakawa. We have seen some resistance in Mānuka and Kānuka. In the case of Mānuka, we're seeing leaf and stem phenotypes, and they're not necessarily connected. For the leaf resistance, we're seeing two forms. So L1 on a modified Jungian scale is what we're terming immune, where we're not seeing any sort of sign of disease. The leaves look healthy. L2 is that hypersensitive response, where we're seeing the localized cell death but no pustule formation. And analysis of the initial phenotype and genotype data suggests that we actually have leaf and stem resistance mechanisms. And again, they may or may not be connected. They could be due to different genes or different gene regulation. And what we're trying to look at is in this classic disease triangle where you have the right environment, the pathogen and the host, and the early stages, how they come together to form disease. So what's happening at the molecular level between Austropuccinia and, in our case, Mānuka. And the tool that we've chosen to study this is transcriptomics. So you've just heard a wonderful talk about that. Looking at the messenger RNA, the sequences that are being shared between the host and the pathogen. And we're not the first to do this. There's plenty of studies looking at the host pathogen transcriptomics for myrtle rust. They're largely focusing on the plant response, and some of this is down to the experiment setup, but also there's been technological limitations as to what you can get from this type of study. There's a study there noting some fungal transcripts, but as I said, not really a focus for this work. On the host side, despite there being studies on different species with different methods and techniques, they do have some common findings when they look at the resistance profiles. And so it's things like transcription factors, receptor-like kinases, R genes that are coming up as the common things providing resistance. And what we're wanting to know is it the same thing happening for Mānuka? And also, can we get into the pathogen side of this story? As set up for this, Louise Shuey, who actually did all of this work in Brisbane on plants that were sourced, or seeds that were sourced from New Zealand. So we had two treatment groups, control and inoculated. We had three groups of plants. So these were phenotyped before the experiment. We had L1, which is that immune-resistant phenotype, L2, the hypersensitive response, and L5 as our susceptible group. And all of the plants we used were stem resistant. So this is a complicated space, so we're trying to simplify it and make it a little bit easier for ourselves. And also the plants are not clones. We have three groups, but they come from different families. We know the mother plant, but we don't know what sort of the other side of the genetics is. So again, still complicated. We've run two experiments. The first one had two sampling points, 24 and 48 hours post-inoculation, so trying to see what we can see at those early points. And then the second one, more detailed samplings, including a pre-spray point. And so for experiment one, when we got that data, we started to look first at the host. And so we're lucky to be allowed access to the Mānuka genome for this work. And we're able to see host genes, so good sign that things are working well. When we looked at the top 100 differentially expressed genes and did a clustering analysis to make this PCA plot, we could get a pattern like this. And what we see is that these samples are clustering based on family, so that's a large driver of the differentiation there. They're also clustering on the other axis by time. So again, we're seeing a shift between the 24 and 48 hours in the plant response. And we're also seeing a little bit of differentiation there based on the treatment. We're also seeing a few things that suggest there may be underlying differences to the leaf surface structure, so that the L1s might be physically different from the L2 and L5s, and that's another area of research that requires some different methods to follow up. And the key question after this was, is there anything from the pathogen in this data set? And again, lucky to have a reference genome to use for this, and we were able to again pull out differentially expressed genes, looking at the top 100 and doing that clustering analysis. Again, we can see two nice clusters in this one. And this time it is, they're clustering based on time. So at 24 hours, we're seeing a really nice tight cluster. So the samples at that point are very similar, and then they spread out in that 48-hour point. There's not really enough sample points there to really say if they're separating very much based on the plant phenotype that they're on. So we didn't see fungal transcripts in the uninoculated plants, so another good sign. And when we looked at the top 6 differentially expressed genes, 3 of them were effectors. One was a putative cellulase, and then a mycotoxin production enzyme and a cell wall enzyme. And so we're really excited to see 3 effectors. They were mentioned earlier. And just to refresh your memory, so a definition we like to explain what an effector is. So it's something that facilitates pathogen entry into the host interior, suppress plant immune perception, and they alter the host physiology for the pathogen benefit. There are 367 of these predicted in the psidii genome, and they're fairly well studied across a wide range of rust pathogens. And we know from the previous work that these effectors are expressed in waves throughout the infection cycle, and they're often timed with other enzymes like hazymes, proteases, lipases, or transporters. So they're actually really key for infection to proceed successfully. When we dug more into effectors in this data set, we saw that there were 16 of them that had significant differential expression over when comparing 24 to 48 hours. This is another clustering analysis. The dendrogram at the top shows that these samples split into 2 groups. And again, it's really driven by those 2 time points, 24 versus 48. The phenotype of the host isn't a big driver here. So thinking here is that in this early time point, the spores land on a leaf and start their infection process, and then it's later on when they work out whether it's going to be successful or not. So by the time we'd done this analysis, we had data from experiment 2. So this was the same setup, but with more time points. And in this case, we had 23 effectors with sequence coverage. Again, our controls looked good, nothing in the pre-spray samples or in the uninoculated plants. Interestingly, so we're seeing fungal transcripts in our 20-minute samples. So as soon as Louise and Bex had inoculated the plants, samples were taken, and there are fungal transcripts in those sequences. For the 23 that are there, you can see there's a range of different patterns of expression. 2 that we're really interested in are these ones. So we have effector 1260, peaking at 24 hours, a sort of higher expression in L5, the susceptible plants compared to the resistant ones. And we're really excited to see this one because we also saw it in experiment 1, so we're seeing some sort of reproducibility within our data. The second one there is 143. We're seeing a peak there in the 6 to 12 hour window. Again, this is one of the ones where we have sequences in those 20-minute samples. So it's there right at the beginning. So now that we have effectors of interest, we are working to characterize them more to find out what they do. So looking at things like the protein structure, so doing work to express, purify, and characterize them. We're looking for interaction partners. So a yeast-2 hybrid library is being generated from RNA from these experiments. It's currently doing the last QC steps, so we're hoping to get that ball rolling really soon, and so that'll be trying to identify what in the plant these effectors are interacting with so we can understand what they're doing. We're also doing RNA assays, so with both detached leaf and whole plants to, again, understand how important these effectors are for infection to occur. So Nikki and Jovan are at the table here, and they have posters later, so please go and talk to them about the really exciting protein work that they're doing on these effectors. And just to summarise, so there are significant challenges working with non-clonal plants. The data is really messy, but I think once you see a trend, you can be fairly confident that it's something real. We're seeing fungal transcription is independent of host plant phenotype, particularly at those really early time points, so again suggesting there's some sort of pre-programmed approach that the pathogen's taking. And again, we're seeing those transcripts for putative effectors in the 20-minute samples, so they're likely preformed in the ungerminated spores and essential for infection. And there's much more work to do to understand the importance of the effectors and to characterise their functions. And that's me. Thank you.
Thank you, Rebecca. It's good to see the genome work that we did has actually got some legs, got moving forward. Yes. Does anyone have questions? Benjamin.
Hi, I'm Benjamin. I had a question. So early on, you see expression of these fungal genes, but obviously you have very little biomass. So how did you actually detect these gene expressions? Did you just sequence a lot? And how many of your reads actually map to the fungal transcripts at 20 minutes?
It's a small number. I think 1% would be the highest, but I think we have the advantage of doing this several years later than other studies. You get more sequence data per run, and it depends, again, how many you fit on a lane. So yeah, it's small, but they're there.